So looking at our first objective in this packet is to describe how urinary excretion of solutes and water influences the volume and composition of plasma and identify other processes that affect plasma volume and composition. So to kind of put this more simpler, the amount of urine that we excrete or that we void is a reflection of how well our kidneys are fine-tuning that filtrate, okay? And so if we can reabsorb things like more fluid and more sodium, that is going to have an influence on our plasma composition, our blood volume, and our blood pressure. So the amount of fluid that we lose via the kidneys and via the urinary system influences things like our blood volume and our blood pressure on the other end. So it's kind of like two sides to the same coin. And so that's what we're going to speak all about today, how the function of the nephron, either reabsorbing or secreting, influences how much urine is lost, and by extension, how much is retained, which influences our blood volume and our blood pressure. Okay, we'll talk about functions of the urinary system. We'll introduce the four basic processes of renal exchange. And then we'll once again talk about the specialization of the renal tubule. So going back to this idea that each segment of the renal tubule, of the nephron even, has a very specific function that is related to a very specific structure, okay? And then lastly, we'll talk about excretion at the very end. So looking at some of the functions of the urinary system here, we kind of introduced this in the last packet and we'll talk about it a little bit here as well. So we're going to regulate plasma ionic composition, things like sodium, potassium, how much of those ions are existing in our plasma, and by extension, what does blood pressure look like? We're also going to regulate plasma volume. Again, how much fluid is being reabsorbed into our plasma or secreted into the urine. And again, that is also going to influence our blood pressure. Plasma osmolarity. And we'll talk about this word osmolarity quite a bit, but whenever we say osmolarity, we're really just referencing the amount of sodium or the saltiness of the plasma, which influences blood pressure. Okay, so osmolarity is a reflection of the solute concentration of our plasma and by extension, our blood pressure. It also regulates plasma pH, so things like the alkalinity and acidity of our plasma, which we will speak about, the fact that bicarbonate ions and hydrogen ions are regulated through the tubular structures, and then removal of metabolic waste products, nitrogen, urea, some of those extra products that are produced as an extension of breaking down things like proteins that come in from our diet. And then some of the other sort of secondary functions of the kidneys are secreting erythropoietin and renin, we talked about just a few minutes ago, activating D3 to calcitrol, so cholecalciferol to calcitrol, so the active form of our vitamin D that can be used in our system. And vitamin D really plays the role of a hormone. Although we say that it is a vitamin, it has a lot of other regulatory functions, and it is closer in its function to a hormone than it is to a vitamin. And then lastly, gluconeogenesis, which is the ability of the kidneys to use other substrates, right, such as fats, to create a new source of glucose that we can tap into during states of starvation. So we talked a little bit about gluconeogenesis last semester, the ability to make new glucose from other substrates such as fats and proteins. Okay, and the kidneys have, along with the liver, have an ability to do that. Just refreshing ourselves of the structure of the nephron here. So we've got our vessels here. So these would be our arcuate artery and arcuate vein, if you remember these two vascular structures coming from some of the major vessels from the renal artery. We've then got our afferent arteriole. So the afferent arteriole is going toward our glomerulus. Here's our glomerulus. And then we've got the efferent arteriole, which is going away from our glomerulus, okay? We then see the peritubular capillaries. And I talked about this last time we met, but the peritubular capillaries are kind of entangled, right? Tightly entangled or wrapped around the tubular structure. So they're wrapping around the proximal convoluted tubule, which is here. So here's the proximal convoluted tubule, right after the glomerulus. And then it also wraps around the distal convoluted tubule. And again, I'm highlighting this relationship that the distal convoluted tubule wraps back around and is nestled in between the afferent and efferent arteriole, okay? And both of these parts of the tubular structures are wrapped or entangled with these peritubular capillaries. And this is what facilitates the ability to reabsorb substances back into the blood or to secrete substances that are no longer needed or wanted, such as toxins, from the capillaries into those parts of the tubule. If we look a little bit lower into the medulla of the kidney itself, where we have the nephron or the loop, this is where we see the vasa recta. And the vasa recta has a similar relationship or a similar association with the loop. Excuse me, sound like Darth Vader there. With the loop of Henle, which dips down into the medulla of the kidney, okay? So this is also how we are able to reabsorb substances, especially in the loop, it's gonna be water mainly, reabsorb that water from the tubular structures back into the plasma via this association with the vasa recta, okay? All right, so let's begin by looking at the exchange process. So sort of adding some layers to that general description. But we really want to bear in mind the relationship of these capillaries to the tubular structures before we get into these processes. So the first of our processes is going to be glomerular filtration. This is where we filter our plasma through the glomerulus and eventually into that glomerular capsule or the Bowman's capsule, okay? And if we think about or look at the structure of the glomerulus, it's pretty much like the sieve-like apparatus where we can squeeze our blood through the sieve, similarly to the way that you would drain pasta in a sieve. And we keep larger substances like erythrocytes and proteins behind in the plasma, but we squeeze all of that fluid right out into that Bowman's capsule. And that's the first part of our process, that is the glomerular filtration, okay? The second part is reabsorption. This is where we move substances that we want. So we may have filtered things out inadvertently that we need, things like amino acids and glucose and ions. And so we want to reclaim those substances instead of letting them being lost in the urine via excretion. And so reabsorption is the initial process of moving those useful substances from the tubular structure back into the vasculature via the peritubular capillaries, okay, or the vasa recta, depending on where we are in the tubule. Another exchange process is going to be secretion. So secretion is where we move substances in the opposite direction. This may be things that escaped filtration. So very small particles like toxins or byproducts, metabolites from medication, or other types of unwanted substances. Let's say we have too much sodium in the blood, right? Let's say we have high blood pressure, we have a lot of sodium in the blood. We can then secrete that into the urine so that it can be lost via excretion. And so secretion is going to be moving substances that we don't need, but that were not filtered out, right? They're still in the vasculature, and we can move those substances directly across from the vasculature into the tubular structure because of this association with the peritubular capillary or the vasa recta. Okay, so this is movement in the opposite direction. Excretion is the final part of the renal exchange process. And some literature will tell you that this is not an exchange process as such, nothing is being moved or changed. And so some books or some texts will tell you that this is an entirely different process altogether. But excretion is pretty much just moving that fine-tuned filtrate from the tubular structures to the ureters, bladder, and then urethra so that it can be voided from the body. Okay, so strictly speaking, there's no exchange happening as a part of excretion. It's just movement, mechanical movement at this point of that filtrate to be voided from the body. And this is what we call urine at this point. All right, so just kind of reiterating that here, we've got our afferent arteriole bringing blood toward the glomerulus. We've got our efferent arteriole sending blood away. We're filtering most of the fluid component of that plasma, right, of that blood, filtering it across the glomerulus, keeping behind things like proteins and cells, which continue to move toward the efferent arteriole. And then we begin to fine tune that filtrate once we enter the first part of that tubular structure. So we start by reabsorbing things that we want that may have been filtered out inadvertently. And then we also secrete things that we don't want that were not filtered across the glomerulus. And then the last part of the process is excretion, where we kind of just void that final substance, which is urine at the very end of reabsorption and secretion. Okay, let's start to look at the first process in a little bit more detail. So this is glomerular filtration. And this is the movement of protein-free plasma from the glomerulus to the Bowman's capsule. Very similar trend to what we saw when we looked at the movement of fluid across our capillary bed, right? Proteins do not get to move across these substances. They are just too large and too useful, really, and they have to stay behind in the vessel, in our capillaries. And so all that is moved across the glomerulus is really the watery component of our blood, of our plasma, okay? Cells, although it's not mentioned here, large cells like erythrocytes also do not make it through. This is why we should never see blood in the urine. There should never be erythrocytes making it across that glomerulus, okay? Now we filter about 125 milliliters per minute, or about 180 liters per day. So that's how much of our blood is being filtered per minute or per day. So a lot of blood. That kidneys are really working overtime to help clear the blood, clean the blood in this way. And the glomerular filtrate must actually cross 3 different barriers to get from the vessel, where it is, the afferent arteriole coming in, over into the Bowman's capsule. It's got to cross 3 different structures which form the filtration or glomerular membrane. Okay, so there's the fenestrated capillary, and a fenestrated capillary is a capillary that has slightly wider or larger pores in between. Okay, we talked a little bit about the different types of capillaries before, and we talked about continuous capillaries, fenestrated capillaries, and then discontinuous capillaries, if you remember that. So fenestrated are the ones that are sort of in the middle. They don't have pores that are as small as our continuous capillaries, but they're nowhere near as large and as sort of free-flowing as the discontinuous capillaries were. We've also got the glomerular basement membrane. So it's really the basement membrane of the glomerulus, right? The basement membrane of that vascular structure. And then we've got the visceral layer of the glomerular capsule, which is composed of cells that are called podocytes, okay? So looking over here, we can kind of see that structure, starting with this panel at the top. So we're seeing the afferent arteriole, which is feeding blood toward the glomerulus. And if we remember describing the glomerulus as basically a ball of capillaries. So we can think of it as being a vessel that's kind of being shoved up or bunched up and pushed down inside this Bowman's capsule. And so the vessel itself has the fenestrated capillaries as well as the typical basement membrane that is associated with our vessels. Okay, that's the first 2 layers of this filtration barrier. The third layer is really a part of the visceral component of the Bowman's capsule. So thinking about this relationship here, where the Bowman's capsule is tightly or intimately connected to the outer aspect of those endothelial cells, those vessel cells, this visceral layer actually has cells that are called podocytes. And podocytes really reflecting the fact that these cells have foot processes. So kind of like these little finger-like projections that have splits, in between here. So if you imagine your hand kind of grabbing onto a structure, the spaces in between your fingers are what these filtration slits are. And so it's just going to provide this opportunity for some substances to pass through the fenestrated capillaries and eventually through the filtration slits that are created by the foot processes of these podocytes, okay? And then from here, it's going to go directly into the Bowman's capsule or the Bowman's space. And this is how we get our plasma to move through these three membranes and eventually end up in the Bowman's capsule. Alrighty. Okay. So here's another intimate look at that relationship. So we've got our ball of capillaries, which is the glomerulus, lined by fenestrated capillaries. We've got our Bowman's capsule, which has two layers. It's got the visceral layer, which is tightly or intimately connected to the glomerulus, and then it's got a parietal layer, which is forming this space where that filtrate can be captured. Looking more closely at the filtration membrane here, we're seeing the capillary endothelium, so the lining of the vessel, which is formed by fenestrated capillaries. We're seeing the basement membrane of that capillary. So underneath that endothelial layer, we've got a basement membrane. And then we're seeing the visceral layer, so the intimate layer of the Bowman's capsule, which is really formed by these podocyte cells. And these podocytes have these foot processes, which create these really large gaps in between that we call filtration slits. And so what we're seeing here is this sieve-like apparatus, this apparatus that allows some things to go through and others to stay behind, one created by the fenestrations of the endothelia, but also by the foot processes that are creating these filtration slits. And so this is what's going to allow most of the fluid to escape, but a lot of the large particles, the cells, the proteins to kind of stay behind. Okay. All right, so I'm going to pause here again and see what questions we have. All right, now we're going to speak a lot about how filtration happens, and we're going to talk about these Starling forces once again. So this should be deja vu at this point, right? It's the same exact idea. We've got 4 Starling forces that are operating in order to move fluid across the glomerulus in a very similar way that it operates in order to move fluid across our capillary beds, okay? So we've got the glomerular capillary hydrostatic pressure, which is represented by this large P and GC. This is the hydrostatic pressure that is being exerted by the fluid within the vessels, right? So glomerular capillary is the location. Hydrostatic is describing the force. So it's going to be a push force from the vessel out into the Bowman's capsule. We've also got the Bowman's capsule oncotic pressure, right? So kind of the opposite force. So this is a force that is created from the Bowman's capsule, but it's represented by the proteins, right? So remember this idea that oncotic pressure is represented by the solutes that are in the region that exert that osmotic pull toward them, okay? So this is going to be a pull force located in the Bowman's capsule itself. So this is represented by that pi sign and BC. So this would be a force that is pulling fluid from the glomerulus into the Bowman's capsule, okay? The third Starling force at play here is the Bowman's capsule hydrostatic pressure. Again, think about where the force is located. It's in the Bowman's capsule. And then think about what type of force it is. It's a hydrostatic pressure exerted by the amount of fluid in the Bowman's capsule. And so that is going to be a push force. So if we're standing in the Bowman's capsule and we are exerting a push force, that would be a force moving back into the glomerulus, okay? So that would be represented by this large P and BC, and that force goes back into the glomerulus from the Bowman's capsule. And then the final Starling force acting here is the glomerular capillary oncotic pressure. This is represented by, again, pi, right? This is an oncotic or a pull force, and this is locating in the glomerular capillary itself, okay? So once again, we have the glomerular capillary exerting a pull force. So the direction of this force is favoring movement from the Bowman's capsule back into the glomerular capillary, okay? And so in a very similar way that we want to think about each of these forces and how they influence the movement of fluid to then determine the overall direction of fluid movement, whether it favors filtration or favors absorption, this is how we will approach the glomerular filtration pressures as well. Now, for the bigger picture, we know that this is going to favor filtration. So we know that our filtration forces are going to be, let's say that again. We know that our filtration forces are going to outweigh our absorption forces. And so that's kind of what we'll go into this with that idea, but we still want to prove that. So we're going to think about looking at each individual force and then calculating our glomerular filtration pressure to then prove that filtration is what's happening here based upon these four forces at play. Okay. I will ask one question here, just sort of as a teaser. So if we think about this idea, which I mentioned earlier, that proteins do not escape the plasma, they do not escape the glomerulus, right? They cannot fit or move across the glomerular membrane or the filtration membrane. And so proteins should never exist in the Bowman's capsule. Based upon that understanding, which of our four Starling forces should always be zero? So take another minute. We don't have many minutes left, but take a minute or so and kind of just think about that idea. So if proteins should never exist in the Bowman's capsule, if they should not be able to move across the glomerulus, which of our four Starling forces should always be zero? Okay, so I'm seeing a response in the chat here that says oncotic Bowman's capsule. So the Bowman's capsule oncotic pressure. And that is absolutely correct, right? Perfect. And again, thinking about the fact that protein should not exist in this space, there should not be an osmotic or an oncotic pull, which is this force here. The Bowman's capsule oncotic pressure should always be zero.